Morning, everyone. Welcome to our Coulter seminar series. Today we have Dr. Georgakoudi. She's a professor in the Department of Biomedical Engineering at Tufts University. She has received her BA in physics from Dartmouth College, then moved on for her PhD in biophysics from the University of Rochester, and performed her postdoc work at MIT G.R. Harrison Spectroscopy Lab and Wellman Center for Photomedicine at Massachusetts General Hospital. Her lab is interested in the development of new and improved methods to assess different aspects of normal and diseased development of human tissues that rely on light interactions with endogenous chromophores and are thus non-invasive. She's particularly interested in the areas where it involves development of quantitative methods to assess cell metabolism and matrix organization features in tissues, and everything is done non-invasively. The main applications of her research from her laboratory include cancer diagnostics, adipose tissue function, osteoarthritis, cardiovascular, and neurodegenerative diseases. She has published over 100 peer-reviewed manuscripts and book chapters in this area, and has also served as program chair, co-chair, or session chair for over 25 international conferences in biomedical optics. She's currently the associate editor for the journal Optical and a fellow of the American Institute of Medical and Biological Engineering at the OSA and SBI. With this, I'd like to welcome Dr. Georgakoudi. She said it's actually snowing out there, although it looks pretty bright and sunny from here. It's pretty deceiving, but she said they had their first snow. And with this, I'll leave it to her. Thank you so much for being here, and I'll let you take over from here.
Great. Thank you. Thank you, Anu. And I thank all the people that have been involved in organizing this seminar. It's really a pleasure and an honor for me to participate in your department seminar series. And I am excited to spend a little bit of time sharing with you our work on using label-free high-resolution imaging to assess different aspects of tissue morphology and function. It probably would have been much nicer if I were there and was able to interact with you in person, but we will do our best. Okay, so all the images and all the results I will show you today rely on nonlinear interactions between light and tissue. So just to make sure we sort of start on the same footing, in these interactions, instead of having a photon coming in, interacting with our specimen and having a single fluorescent or scattered photon out, we have two photons that are coming in that are interacting with our molecule at the same time. And that leads to a single photon that's fluorescent or scattered from our sample. Since now we have two photons that together provide the same energy for this interaction, the wavelength of the photons is much longer than the corresponding single photon interactions. That means we can use light in the near infrared where scattering is much less than in the visible. So that's really nice for imaging techniques because it means that we can go deeper into the tissue. However, these nonlinear interactions are much less probable than your linear interactions. That means they require a very high density of photons. And to achieve that high density, so to make those probable and detectable, we focus the photon density in time by using lasers that emit light in femtosecond pulses. So we can have our peak photon density very high, but maintain average power levels that we illuminate the tissue with to milliwatt levels. So we are obviously not damaging our tissues. And then, of course, we also focus our light in space. Now, the beauty of these nonlinear interactions and the need for super high density is nicely visualized here, where you can see if you can come in with a laser beam focusing into this fluorescent solution, you get your highest density, obviously at the focus, but you create fluorescence both above and below the focal plane. Which is okay, but you need to remove that background signal if you're doing confocal, for example, because you just care to get your signal from the focus, or you have a confocal pinhole, and that makes the process lossy. You can get photo bleaching, photo damage in regions where you're not imaging. In contrast, when we come in with our near infrared beam of light, we get enough density of photons to just get our fluorescence generated at the very focal point of that beam. And now we know all the signal that's generated is coming from the location that we're interested in imaging, and we can scan that spot and get our high resolution 3D images with higher detection efficiency and less photo damage in principle. Let's see. Okay, so the main point that I want to make through the examples that I will show you is that label-free two-photon microscopic imaging. So I should say that also all the images that I will show you from my group are images that are acquired without using any exogenous contrast agents, including these images, for example. And the main point I want to drive is that we can really use two-photon imaging in combination with a natural, the organic contrast that's present in our tissues to get a wide set of unique metrics of tissue, metabolic, and biomechanical function. So we're not going to just get pretty images and morphological information, but our goal is to really use that information to understand what's going on in the tissues, because I think that's a great way to really drive improvements in diagnosis and therapy in many diseases. So metabolic function and biomechanical function are the two sort of functional properties of tissue that we focus on in our studies. And I will show you how, again, we can look at NADH and FAD, autofluorescence, and get metrics of metabolic function, and then look at the natural nonlinear scattering or second harmonic generation and the fluorescence coming from collagen. And then again, get assessments of how biomechanical properties change. Now, why do I care to look at both cell metabolism and biomechanical function of primarily dictated by collagen? Collagen is the dominant protein in our extracellular matrix, and it's becoming more and more obvious that for normal tissue function, development, and conversely, whenever disease develops, it's not enough to just think about the cells or to just think about the environment or the matrix in which the cells reside. It's really the combination of the two, their communication and their interactions that ultimately play a key role in whatever is going on. So our goal here is to use these tools and really get a handle on these interactions and how they affect each other. So cell metabolism, as I mentioned already, is the cell functional property that we're interested in characterizing and understanding. Cell metabolism is a broad term that refers to a set of pathways that the cell utilizes to produce energy and synthesize molecules. Take your favorite disease. It's highly likely that there is a metabolic dysfunctional component that's associated with it. And while we do have tools to assess metabolism, we definitely could use tools that enable us to assess better metabolic function and dysfunction, especially at the cellular and the subcellular level in a non-invasive way. Now, mitochondria are organelles in our cells where typically most of the energy is produced. And what is really neat about mitochondria is that they naturally fuse and fission throughout the life cycle of the cells, as some of these videos show, in response to stimuli, in response to treatment. And this is what the cell does to actually produce and deliver energy in a highly efficient way. And I will tell you a little bit about a technique that we developed that's particularly well suited to characterize some of these dynamic organizational changes in mitochondria. We do have tools, as I said, to assess metabolism. In the clinic, techniques such as PET and fMRI are used routinely to assess metabolism and help us detect tumors, metastases, brain activity routinely. In the lab, especially the last 10, 20 years, there's been an explosion in the area of metabolomics that has been fueled by some really nice improvements in mass spec, in mass spectrometry instrumentation. These techniques are, of course, highly invasive and destroy the sample. Techniques such as fMRI and PET, I should say, are obviously techniques that we use in humans already in vivo. A lot of the times they require contrast agents, and the resolution is at best on the order of hundreds of microns, if not millimeters. So we can't really see individual cells. In the lab, to visualize metabolic function and mitochondria in particular at the cell level, there are a number of stains that have been developed and continue to be developed. But again, thinking about transferring these to animals and especially to humans is not trivial. So what do we know about metabolism? We know that metabolism and metabolic changes are highly dynamic. And the rate of change of metabolism can be very fast. So things can change on the order of seconds, actually even milliseconds, or things can change over months and years. And both types of metabolic changes are important to understand and characterize. Metabolic changes are also highly heterogeneous. These are examples of images from populations of cells that are in principle or started as the same population of cell treated with the same agents, yet cells that are right next to each other respond and function metabolically very differently. So clearly, images of this resolution you cannot get with PET and fMRI, and this sort of changes in humans and tissues over time you cannot assess on the same specimen with something like mass spec. So how are we doing it? We rely on the natural fluorescence that's coming from NADH and FAD. Those are coenzymes that are involved in a number of metabolic pathways, and we know their two-photon action cross-section and their emission characteristics. We've known those actually for a while. There's also some unique fluorescence intensity lifetime decay properties for these two molecules. In the case of NADH, when NADH is bound in the mitochondria, the fluorescence lifetime increases quite a bit versus lifetime of NADH when it's free in the cytoplasm. We've known that NADH and FAD naturally fluoresce for over 50 years now, actually close to 60, 70 years through seminal studies that were performed by Burton Chance, who by many sort of considered the father of biomedical optics. And he relied on these fluorescence changes to actually look at metabolism in the kidney and the brain. So how come this is not a tool that our biochemists and biology and medical friends and colleagues don't use routinely to assess metabolism. One of the reasons is the following. Typically, when I started thinking about this, 100 years ago, we thought about metabolism in the context. It's hard to make jokes over Zoom. I don't, we'll have to, anyways. We think about producing energy through oxidative phosphorylation in the mitochondria and through glycolysis in the cytoplasm. So that looks fairly reasonable to try to model and understand and follow, but this is what's really going on. So the set of pathways is fairly complex, and it's really hard to just think about NADH and FAD fluorescence and make sense of the data in a really meaningful way. So many metabolic pathways besides oxidative phosphorylation and glycolysis can affect NADH and FAD fluorescence. Can we still use them to get information that's meaningful and can help us improve our understanding of tissue function and disease? And of course, I think that's the case. The way we are trying to address this problem is by taking on a sort of multi-parametric approach. So let's see what different aspects of metabolic function can we characterize using these endogenous signals. One of these metrics of metabolic function that we utilize is this optical redox ratio that we define as the ratio of FAD autofluorescence to that of NADH and FAD. So we can acquire images of NADH and FAD, and for every pixel in our images, we can have a redox ratio and display redox ratio coded images. We've shown that this metric actually correlates quite nicely with corresponding mass spec measurements. So we know we're measuring what we think we're measuring. The second metric that we use relies on this fluorescence lifetime and the differences in fluorescence lifetime between bound and free NADH. Typically, lifetime is described via a sum of exponential decay functions. More recently, actually, a lot of researchers have tried to adopt a Phasor approach. So you basically transform your data into the frequency domain. You don't do any fitting, but you basically look at in the Phasor space every decay pattern, every pixel corresponds to a dot in this ellipsoid. If you have a fluorescence molecule that has a mono-exponential decay, then we would actually see that sort of residing along these universal semicircle. When you have a bi-exponential decay, as in the case of NADH, then you end up with points that lie along a line that cross this semicircle at points that correspond to the long and short lifetimes for the bound and free NADH. So what we do actually is we use some very simple math. We look at the centroid of our ellipsoid ultimately, and we use that as a metric of NADH bound fraction. And now we can color code our images as our NADH bound fraction. So that's a second metric. The third metric is our mitochondrial clustering metric. This metric relies on the fact that when NADH is bound in the mitochondria, its quantum efficiency of fluorescence goes up by tenfold. So one of the things that we assume is that the intensity fluctuations that we measure in our NADH images are actually representative of the presence and absence of mitochondria and the organization of the mitochondria in the cell. So we've come up with a set of approaches that basically enable us to take the NADH fluctuations in the cytoplasm, clone stamp them on square images, and then use a Fourier transform approach to get the par spectral density or the squared amplitude of the Fourier transform, and then fit that to a very simple inverse bar law expression, and use the exponent of that fit to give us a metric of mitochondrial organization. So these are examples of images of cells that whose mitochondria have actually been stained and visualized more easily. And you can see as mitochondria go from a more sort of networked to a more fragmented state, these exponent increases. So the mitochondrial clustering metric goes up. This is a metric that we've actually validated even in humans from images that were acquired in vivo in a study that was done in collaboration with Bruce Stromberg, who was at the time at UC Irvine still, and Mihaela Balu, who's still there and with whom we're still working. So these are NADH images that are acquired from a human forearm while the person is living and breathing. These are images of the basal layer of the skin. A hypoxia is induced in the patient by occluding arterial flow. We see actually NADH fluorescence changing very soon after the onset of occlusion, and along with those changes in NADH intensity, we see actually the clustering of the mitochondria also increasing significantly and actually staying increased much longer than the NADH intensity after the occlusion is terminated. So we have these three metrics, and we can extract these metrics using the same instrument at the same time. And we have done that for a set of studies in which we introduced actually a series of perturbations in some of the major metabolic pathways, including glycolysis, glutaminolysis, uncoupling, fatty acid oxidation, and fatty acid synthesis. And what we see is that if, for example, we look at just a decrease in the redox ratio, then that can be caused by perturbations in a number of pathways, including glycolysis, fatty acid oxidation, and fatty acid synthesis. Well, when we look at it in combination with NADH band fraction, sometimes things change the same way, sometimes things change in opposite ways. And then what's more importantly, when, again, we put all of these three together and we look at both at the direction and the magnitude, we find actually to our very nice surprise that each one of these perturbations ends up occupying a fairly unique space in this 3D volume. So what this is indicating now is that we can look at a combination of changes in these three metrics and go back to this map and sort of use it as a guide to get some insights regarding what metabolic pathway, what changes in what metabolic pathway is actually leading to the changes in the fluorescent signals that we are detecting, and hopefully use that to understand a little bit better how a particular disease develops or what may be better ways to treat it. So I'll show you some examples of how we use this to improve diagnosis and or understanding. So the first example I'll show you is from a study that we recently published that focuses on the use of label-free two-photon imaging to assess, to identify precancerous lesions in the cervix. The human cervix is a squamous epithelium, so there are many layers of cells at the surface. And as a result, typically of human papillomavirus infection, there are some changes, some genomic changes that then lead to morphologic and functional changes that occupy ultimately, when you get to the high grade squamous intraepithelial lesion state, the entire epithelium. And then ultimately you have invasion and metastases. Thankfully, most cervical cancers are detected at the precancerous state. If you get to the invasive state, actually cervical cancer is quite lethal. Worldwide, it's actually the second leading cause of cancer death in women. In developed countries like the US, where we have better screening tools, it's not as deadly. However, limitation here is that out of 100 biopsies that we take from women that have suspicious lesions in their cervix, only 10 of them are likely to harbor these high-grade lesions. That means that these patients need to be treated. The other 90 are low-grade or benign lesions, which means the patients need to be just followed up. At the same time, we know that our sensitivity of detecting a lesion goes up from 60% to nearly 100% as we go from 1 to 2 to 3 biopsies. The standard of care for these cervical precancer patients is actually to take a single biopsy. And the reason for that is, one of the reasons for that, at least, is that these biopsies are taken under no anesthesia. Women are tough. We're supposed to deal with pain, apparently. So bring it on. That's the standard of care. Local anesthetics actually do not work particularly well to alleviate pain in this case, but there's definitely room for improving the care to these patients and our ability to detect and diagnose these lesions better. So these are actually optical sections through the epithelium of freshly excised human cervical tissues. Whoops, there's a normal, it was a normal, sorry. There's a normal, there's a low-grade lesion, and there's a high-grade lesion. I think you can very nicely see the cells. You can see, especially in the case of the normal, you can see the significant changes in morphology that occurs as a function of depth. Things look pretty, but they don't change as distinctly in our high-grade and low-grade lesions. And it's also really interesting to see these heterogeneity that occurs at the cellular level, especially in our low and high grade lesions. So we've taken the stacks, we analyze them as a function of depth. In the healthy tissues, when we look at mitochondrial organization, we see sort of this very distinctive checkmark pattern. This is something that we also saw in skin of humans that were imaged in vivo. That nice variation and depth dependence goes away when we have the low-grade and high-grade lesions. And here you can see the statistics from the stacks acquired from a number of patients and regions of interest that were imaged. And you can see, again, very significant changes that occur both, actually, this is a metric of the variability as a function of depth. And we see very significant changes occurring at the low-grade stage. These are corresponding images and metrics that are associated with the redox ratio. So we see a significant change in the overall redox ratio across the epithelium at the high-grade level. These differences are not significant yet for the low-grade lesions. Nevertheless, when we quantify the variability of the redox ratio as a function of depth, then we can get significance even for the low-grade lesions. So these metrics of metabolic heterogeneity are very, very interesting and can be very important. And it's certainly something that we're not able to look at clinically. So then we can get a combination of metrics, and actually some of the metrics that we used relied on the morphology of the cells and the thickness of the epithelial tissues. But when we use different types of diagnostic algorithms to see how well we can separate our precancerous lesions from our normal tissues, and then our high-grade lesions, again, this is important because these are the patients that ultimately need to be treated. From the non-high-grade lesions, we get very, very high sensitivity and specificity, and we have just embarked on a study to actually, well, of course, continue acquiring ex vivo data, but really build a system and try to do this in vivo. I'll just show you one more example from a completely different set of specimens. These are engineered brain tissues that are built on these silk hydrogel scaffolds that are embedded with different types of matrix, and then glioblastoma cells from patients. We see, and these are redox coded images, we see metabolic differences in the cells grown in the scaffolds, depending on whether the cells are residing out here or further inside the scaffold, as you can kind of tell from the hues already here. And of course, we can see some nice differences in the changes that occur before and after we treat with TMZ. And again, it's interesting to see that the sort of the scale of the change is different, whether the cells are at the outer edge of the scaffold versus the inner edge of the scaffold. Again, this heterogeneity, very important, potentially not just for diagnosis, but also for understanding treatment responses. I was going to tell you about fat, but I think in the interest of time, I am going to skip my fat slides and maybe just get to the point. So it turns out that fat has and lipid, well, adipocytes actually have beautiful autofluorescence, and the lipid droplets inside fat fluoresces. And the signatures of that fluorescence, both in the cytoplasm and in the lipid droplets, are distinct, whether we have white fat, brown fat, or beige fat. And we've done an in vivo study with a mouse model where you basically activate your beige fat and you make it consume energy, and we can identify unique signatures again in the metabolic function. And these are you know, a snapshot of the results. Again, when we look at our three readouts, we can really separate quite nicely the brown fat from the beige fat, from the white fat, especially after we activate it with cold. And we get, again, the brown fat that we have lots of when we're babies to produce heat, actually gets activated, burns energy, and again, produces heat. We see that happening in the beige fat of these knockout mice that we used, and actually not much happened as a result in the white fat. So we can use that to potentially monitor what goes on and identify these distinct depots. And the last example I'll show you is actually from an osteoarthritis study. Again, cartilage is the tissue that lines our bones and is present in the joints to provide some sort of cushioning as we're moving around. And cartilage has the surface of the cartilage has three distinct zones. And obviously, as the cartilage sort of degrades, we get osteoarthritis. The problem is that obviously, usually we detect osteoarthritis when we get to the point where there's pain. They're not, even in our animal models, they're not really good tools to help us study the development of this process at very early stages. So we wanted to see if we can use our tools to get some insights. So these are images from these mouse knee cryosections that are coming either from actually primarily sham surgery. So these were animals that were exposed to, basically their knee was opened up and was closed, or a surgery that destabilizes the medial meniscus. So this is sort of a standard osteoarthritis induction model. These mice at about actually 10 to 11 weeks post DMM surgery, they start feeling pain, and we start to notice differences in the way they move around. The samples that we looked at were at much earlier stages than pain, starting from 1 week and going all the way to 10 weeks. So these are examples of the fluorescence images that we acquired from the cellular components, from the collagen. We also looked at second harmonic generation. So this is nonlinear scattering from collagen. And then we looked at corresponding lifetime images. We'll talk about collagen a little bit later. In terms of the metabolic readouts, one of the things that's really interesting is that in that superficial layer, we see metabolic changes that are happening very early, within a couple of weeks from surgery. And the other thing that is really exciting is that while there's one type of difference between our sham and DMM mice, this sort of metabolic difference reverses at the later time changes. So things are clearly changing very dynamically at the surface. We are not detecting significant differences in the deeper layers, not yet at these stages anyways. So again, we can look at the combination of metrics that we have, and we can sort of see, is shown here in the gray, to see what our osteoarthritis tissues are doing. And if we follow sort of how we move in the space, it looks like at the beginning we may have some enhanced oxidative phosphorylation, but then at the later stages we may have enhanced glycolysis and fatty acid oxidation. Okay, so enough about metabolism. I want to spend a little bit of time telling you a little bit about collagen, which is, as I mentioned, the dominant protein in our extracellular matrix. And definitely a number of studies have shown that it's the collagen fiber density, the alignment, the thickness, and the cross-linking levels are all impacting the biomechanical function and properties of that matrix that the cells continuously sense and respond to. And again, luckily for us, collagen has beautiful second harmonic generation and autofluorescence that we can utilize to assess its content, organization, and cross-linking. So we get these beautiful three-dimensional images, and when we started working on this, we realized that they're really not particularly good tools to enable us to exploit the fact that we get images in 3D and get characteristics of organization of collagen in 3D. So we published a number of papers in which we developed the formalism to assess collagen orientation in 3 dimensions. The approach is based on a summation of weighted vectors. And the metric that we ultimately extract from characterizing basically the angles of orientation of the collagen fibers is these directional variance. Directional variance is basically, it's a simple metric that is equal to 0 when all the fibers are aligned in 3D space perfectly, and it is close to 1 when the fibers are completely disoriented. So we've used this metric, for example, to characterize the organization of the collagen fibers that are surrounding these mammary epithelial structures that are embedded in a collagen gel and are treated with different hormones. And again, we characterize the organization of the collagen fibers within about 40 microns surrounding those epithelial organoids in response to the different treatments. And what we see is that actually the directional variance is different. So the alignment of the collagen fibers very much depends on the hormonal treatment. And again, really interestingly, we're able to pick up these differences significantly when we look at the organization of the fibers in 3 dimensions, when we use corresponding metrics by analyzing the individual 2D optical sections. The trends are there, but the differences are not significant. So this is indeed a much more sensitive approach to detecting especially subtle collagen fiber changes. And we can do all sorts of different things. So for example, we can detect or we can characterize collagen organization in different regions surrounding the epithelial structures. And again, we can see differences when you treat with E2 and prolactin, actually, depending on whether you're looking at collagen organization along sort of the long edges of the epithelial structure versus sort of the leading and trailing edges, and so on and so forth. So we can get very sensitive, very detailed information about organization of collagen fibers, looking at second harmonic and analyzing those signatures. Fluorescence is also important, again, depending on the problem we're looking at. This is an example of a study coming from decellularized heart matrices. From rats that were either healthy or had a heart attack, and we're looking at them at different time points following that heart attack. And we characterized in that case both the second harmonic, and we saw that the second harmonic signals correlated quite nicely with the overall collagen content. But when we looked at actually the mechanical, the elastic modulus of these decellularized matrices, and compared the infarcted versus non-infarcted hearts, we saw that the metric that was most correlated with biomechanical function was actually the label-free autofluorescence associated with collagen associated with the crosslinks. So in this case, cross-linking seemed to be a defining factor in terms of the biomechanics of the tissue. In other cases, we see that sort of changes in both the content of the collagen, the organization of the collagen, and the collagen cross-linking are associated and correlated with changes in stiffness. So these are results that are coming from engineered tissues that are prepared using fibroblasts that are coming either from normal human skin or the skin of patients that have scleroderma. So you have stiffer stromal stiffness in these tissues, and that is again correlated with all these nice metrics of collagen that we acquire. Osteoarthritis is the last example I'll show you. As I mentioned at the very beginning, we also looked at different metrics of collagen organization and cross-linking. Again, what is interesting is that especially in the lifetime, we see changes that are quite significant even for the transitional and the radial zones, or the deeper zones. Again, very early we see changes in all of these metrics that are happening in the superficial zone. And in this set of studies, we actually have some nice metrics of both collagen organization and cross-linking, which change actually within a week of surgery, so super early. And then also metabolic function. Again, these changes become significant usually a couple of weeks later. So do metabolic changes impact collagen organization? Does collagen organization impact metabolic function? These are going to be, I think, really interesting questions that we hope to address. So I want to summarize. I've tried to show you that we can extract a combination of metabolic readouts from our cells and our tissues using label-free two-photon imaging. And we can get not only sensitive, but very specific insights regarding metabolic changes and the nature of metabolic changes that are happening as tissues develop or as diseases develop. When we look at a combination of label-free second harmonic and two-photon excited fluorescence images from collagen, we can also get a nice range of metrics of biomechanical function. And then I believe where it's exciting is actually combining the two types of metrics and understanding the dynamics of those interactions. I think these tools are really well suited to address in a unique way questions like this. I think overall, we're getting to a really exciting point to really start thinking about translating a lot of the things that we've been doing in the lab for a while to the clinic, and enable really a paradigm shift where instead of just having our tissues brought to the microscope and then look at them under the microscope in some lab, we really bring the microscope to the patient and get not only the morphology that the pathologists are so used to looking at for their diagnosis, but you look at a wealth of metabolic and functional metrics that I think can really improve our understanding of what's going on. So I should, of course, thank the students and a wonderful set of colleagues and collaborators of all over the place that have been instrumental in doing this work. Actually, most of this work that I talked to you about was done by Zhiyi and Dimitra, both of whom have moved on. Zhiyi is now a professor at Zhejiang University, and Dimitra is a fellow MD, a pathology fellow. This is the gang who's taking over, and of course, some much appreciated funding from a number of sources. So thank you very much for your attention, and I'll be happy to take questions or hear how you guys deal with questions and answers. Oh, I see.
Thank you so much, Dr. Georgakoudi. That was an excellent talk. We learned a lot of new things, I'm sure, and there's a bunch of questions for you. I'm going to just go ahead and start off with what I see in the Q&A session. The first question comes from a PhD student, and he wanted to know, from the tissue samples that are excised from the cervix, are the cells still alive? Are they still metabolically active?
Yeah, no, so this is a great question. Yes, the answer, so we take great care. So there are a number of studies that we've done, and a number of studies that that have been published with epithelial tissues that basically show that for about 4 hours after you get tissue out of an animal or a human, you see metabolic activity maintained. So when we take our measurement, I mean, we take our measurements typically within an hour after the tissue comes out of the body. And that has taken quite a bit of organization and collaboration with the colleagues and the pathology people at the Tufts Medical Center. You put it in the microscope, you know, we're able to image for about 2.5 hours, and we don't see really significant differences in our redox ratio or mitochondrial clustering. After that, we are definitely start seeing the tissues degrade and metabolic function deteriorate. So it's important to do that very quickly after the tissues come out, and make sure that the tissues are maintained like during transport in ice and all the good stuff.
Thank you. We have a question from Dr. Romila Rahman. Is the redox ratio calculated on a single image or depth, or more than one?
So again, for the cervix, we calculate the redox ratio over each one of our optical sections. So we can get a redox ratio that's an overall average redox ratio of the epithelium. But the changes that are more diagnostic are the changes that basically tell us how much the redox ratio varies as a function of depth. And I'm not sure this is answering the question, but maybe I can also chat with, well, actually, I don't know who answered that question, but did that answer
that
the
was
question?
Dr. Romila Rahman. So
Okay.
I don't see a follow-up, but there's one more question. Would it be more to detect on the surface in part due to the challenges of imaging deeper? This is from a grad student.
Yeah, no, I mean, this is a great point and a great idea. And it is definitely something that we're thinking about. For sure, if we can come up with ways to do this and do this well, just looking at the surface, I think it will be fantastic. Both it would be easier, it would be faster, but also if you think about developing tools that are cheaper and easier to use and deploy in countries in third world, then absolutely. I just, yes, we'll have to, you know, again, the depth dependence is definitely diagnostically very helpful and very useful. Teasing out the details of what we can get away with, I think will be interesting and important for sure.
I have at this point 2 more questions. Actually, they're increasing. Let's start from another PhD student. Is how is the variability as a function of depth of mitochondria related to the organization?
Okay. Let's see. How is the question, one more, I actually don't see that question on
this is in the chat box. So this is, how is the variability as a function of depth of mitochondria related to the organization?
Right. So what happens typically in a normal epithelium is that in your basal layer, so in that bottommost layer of cells, you have these are proliferative cells, and typically when you have a proliferative cell, that cell actually relies quite a bit more on glycolysis than oxidative phosphorylation to produce energy. It's a bit of an oxymoron, because you would think this cell is proliferating and needs more energy. Why is it using glycolysis? It's less efficient. But it turns out when you produce energy via glycolysis, you produce molecules that are available for biosynthesis much more readily, and the energy sort of much more readily available to the cell. So that is known. Now, so when the cell decides to produce energy via glycolysis, then the mitochondria don't play as a significant role, and they don't, you know, so it doesn't matter if they're networked and they talk to each other. As the cells move to, so the mitochondria tend to be sort of more clustered and isolated, and that is what we call a fragmented mitochondrial state, and the mitochondrial clustering is high. Now, as the cells sort of differentiate and get to more superficial layers, you start relying quite a bit more on oxidative phosphorylation, and the mitochondria become more networked. Your mitochondrial clustering goes down, and then you get to a point where in the squamous epithelial tissues, the cells sort of start to become senescent because they eventually slough off. And then, you know, your mitochondrial clustering is starting to go up again. And I hope that answers the question.
Okay, I have two more questions real quick.
Okay.
Is Dr. Romela Roman, have you looked at the elastin organization?
Yeah, not yet. I mean, elastin is beautiful. We are just starting to really focus more on collagen and elastin. Our initial studies were primarily looking at the surface. We managed to get a better, you know, we have a better microscope system so we can see even deeper. And we are definitely starting to look into elastin, which is very interesting, potentially very important as well.
Okay, last question from another faculty, Dr. Pullagurta, is what is the projected power requirements for a portable system if this was the most miniaturized and efficient design? Is it in few watts or hundreds of milliwatts?
So the power that we deliver to the tissue is 10 milliwatts, 5 to 10 milliwatts for the surface. If you want to image deeper, you need to deliver at the surface about 60 milliwatts. You know, if you want to go a couple hundred microns deep, and so this is what we're working with right now. It may be possible to do this with even less power. You know, we're starting to think obviously about machine learning and using algorithms like that to do denoising. So, you know, it may be possible to use less power, get images that are of lower signal to background ratio, but, you know, we're still able to analyze them and get useful information, but on the order of tens of milliwatts.
Got it. Thank you so much for answering all the questions. That was our last question. Once again, we'd like to thank our speaker, Dr. Georgakoudi,